Gila for inviting me here. I must say I'm not a genomics guy or a genetician. I'm more interested in very, let's say, earthy things like wine and vines. And this is how we started this project. I'm from the Department of Agriculture and Enology Research in the Samaria and Jordan Regional Rift in Ariel University. And we started with, let's say, an initial work trying to find and characterize Israel's native grapevine population, which was very, very small to start with, and I will explain in a few minutes why. This is actually the first part of what I'm going to speak, and the second part will be more about the archaeological DNA or our goals in this area. There are a lot of collaborators. Some of them are from Italy, giving me all of the, let's say, the help with the genetic part and the archaeological parts. So the question we asked is, where did the Israeli wine varieties disappear? Where did they go? We actually, when we come here after many years of the diaspora, we found only table grapes, and I will explain in a few minutes why. It's known that the wine and the grapes and the raisins and all of these very important commodities were embedded strongly in the culture, both of the Jews and the nations around here. They were a very important part of the diet. The wine was very, very much abundant. You could see wine presses all over Israel, and it all stopped or started stopping in the 7th century. What happened was that new regimes with Muslim religious came in, and they actually oppressed the wine industry because of religious reasons. They are not allowed to drink wine. So it was like, in the first it was moderate oppression, it was not total oppression. But during the 13th century came in very zealot Mamluk guys, and they actually made a decision against wine very strongly, and they started actually destroying the wine industry, destroying the vines, destroying the vineyards, breaking down the wine presses. So from this time on, there was a negative selection, a very strong negative selection against the grapevine varieties that were used for wine. Just for a short explanation, a table grape that we are used to eat is usually have a bigger berry. It has a soft skin. You don't want to actually chew it too much. It should be fleshy. It should have moderate amounts of sugar and acid, not too high. On the other hand, if you want to produce wine, you need to have high sugar levels. You have to have high acid levels. You want to have a lot of polyphenols in the skin. So there are a lot of different genetic characteristics for table grapes and wine grapes. And what we found here today, in modern times, when we come back to Israel after so many years in the diaspora, we found, and people wanted to try and produce wines again, and they were looking around and they saw only table grapes, no wine grapes, and we just explained why. These are the guys in charge for all this mess, the Mamluks. This is a Mamluk knight. So we starting with the Baron Rothschild, the renewing industry in Israel. He goes into Rishon LeZion and makes the first winery that you can see up here. Okay, people working in the renewed wine industry. He brings specialists for France. They look around, as I said, and they see unfavorable varieties, so they start bringing from Europe their own varieties, the Carignan, Cabernet Sauvignon, and so on. The result is, up to this day, there is no wine production in Israel from indigenous varieties. All of the varieties that are used for wine production in Israel today are from Europe mostly, some of them from America, but mostly from Europe. More than 60,000 tons, mostly Cabernet Sauvignon, Carignan, and Merlot, that were brought by the Baron Rothschild officials. Okay, so up to this day, it's a very, very narrowed population. So the question is, can we reestablish our ancient wine varieties and our ancient wine practice, actually? Because the style of wine depends on the variety. So what we're trying to do now, and I will explain, is locating and characterizing our local grapevine population here today in Israel. We are recollecting. The hypothesis was that some of these varieties at least survived in the wilds, because the grapevine is a very versatile, very strong species, and it can survive in a lot of different conditions. Understanding the connections between the wild and domesticated populations in Israel, we found a large, very large Vitis vinifera sylvestris population in Israel. It's huge, and we are characterizing it genetically and by morphology. To identify, this was actually my main goal, okay, as an enologist, to identify varieties that are suitable for quality wine production today, varieties that are ancient and we can actually introduce back into the industry. And trying to close it all, which is why I'm here, through archaeological findings, trying to understand which of these varieties that we actually recollected from the wilds may have been used for ancient wine production. So the collection map, which consists of more than 330 different accessions today, comes from the northern border of Israel, from the Dan, down to the Thousand Negev. So we found actually living varieties of Vitis vinifera down in the total dry Negev, and they are unique species, which is very, very important, because on the next stage we will want to look into them and see if we can find resistant to drought and salinity in these specific accessions. But you can see that the sylvestris lines cluster in the north of Israel up to the Kinneret. The sativa is more spread down all over Israel, even in the mountains and on the seashore. A lot of people know around Palmachim and Nitzanim there are a lot of grapes where some of them are unique varieties, specifically indigenous to Israel. If we look into the sylvestris population, there's as I said, there is a very big clustering of it around the north. Now we know that all of this is full too. We are finishing the survey now. But what we found is a very interesting thing, that there is a stop of a spread of the sylvestris lines around Nachal Semech in the Kinneret. And we are now hypothesizing that the reason for that is salinity in the ground. So we think that the sylvestris lines are not very stable to salinity. They are not resistant to salinity. And we are going now to check it by taking samples from the ground and water around there and trying to understand. So we found sylvestris only up to here and not at all on the western side of Israel, only on the eastern side and on the up to the Kinneret. It's divided this way. The difference between the sativa and the sylvestris are in the flower. The sativa are hermaphrodite. They have male and female organs in the same flower, as you can see here. And the sylvestris lines are female or male individuals. So we now have many more than what you have, what I have presented here, and we are now genetically analyzing them as well. As I said, we want to understand if we can produce wine from these varieties. So some of them make very good wines. You can see samples of wines done from these varieties here. And what was very interesting that we found, you see, this is Bituni, is one of the local table grapes known that were remained here, and it has a formal name. Some of them are very, very ancient, traditional, historic varieties. Some of them are dated 2,000 years ago, with historic evidence for their names going down in history, like the Bituni. Some of them from our collection. So you see the Bituni is not very suitable for wine production. It has about 18% sugar content, very high pH, which is not very good for wine production, and very low TA, total acidity levels. On the other hand, you can see here a collected sample of black grapes, unknown, with no name for now. We have to give a lot of names now, with a lot of sugar, a very good pH level. So there are very good candidates for modern wine production in this population. A very interesting thing we found that the Silvestris, which are not considered suitable for wine production, in Israel, there are many Silvestris varieties that are suitable for wine production. And that's a very important fact because it may set us into a direction of domestication in Israel, because there is a reason for domestication, okay? Because you can see that the amount of sugar and pH are very good for wine production from the Silvestris. And we produced wines from the Silvestris, and they were tasted by specialists in Israel. And they say, look, this is a very good variety. I would like to have it in my vineyard. This may mean for us that Israel was indeed a suitable domestication point. This is the population analysis done by 22 SSR analysis. You can see that most of the sativa population is clustered together. There are some Silvestris lines go, let's say, close to these clusterings, but the Silvestris are divided more or less into three groups, or 2 or 3 groups. We're still not sure. They are geographically connected. So Silvestris from the very high north are one population. Silvestris of the Golan is one population, more or less, and the Silvestris of the Kinneret region is more or less one population. You can see here the structure analysis. The best grade or score we got for a division of three groups, and there is a good score for a 5-division group. I do believe that this is the right one because of the different Silvestris populations that we found, and this explains the 5-group structure analysis result is better. We can explain better the populations of the Silvestris by this division. When putting this on a higher scale of around this all of this region, we saw that we are we say in Hebrew, Am levadad yishkon. We are separated. So our Silvestris population is totally separated from the rest, from the Silvestris of Europe and from many other European varieties and populations. Indeed, even from the Near East, near Asian regions. And in general, some of our sativa are similar to or close to European groups, but some of them are totally separated. So now we're trying to understand on the next step, and I hope Gila will help us with that, to understand, is there an in-house domestication in Israel? Does maybe some of our sativas came or were derived out of Silvestris lines? We even found in Israel something that is very unique. We didn't publish it yet. It's a white Silvestris. It's a common, let's say, knowledge, or it was accepted that the mutations for white grapes happened much later in domestication. But we found a white Silvestris. So the question is, if it's true, or maybe indeed people domesticated white and red grapes and then they spread out. It's a good question. Later on, and this is already in our goal to go further into full genome sequencing of archaeological DNA, we took 10 of our accessions and we full genome sequenced them on 20x, and we tried to find SNPs that explain the most variability in these lines, and after that we clustered them, and we saw that they cluster very nicely. These are Silvestris lines clustered together very differently from the sativa lines. And the sativa lines, the collected lines, the ones that we found around Israel that were not known, not traditional, cluster together. And the traditional sativa lines cluster together with the white ones here and the black ones here. The black one, there's just one there. So this was like a feeling that we are in the right direction. And these 40 high polymorphic SNPs will be used later by us to analyze the archaeological findings. Now, this is more, for now, more questions than answers. We are very much in the start of this work, and we want to try to collect samples of archaeological sites, of grape seeds, and full genome sequence them as possible, and try to find out if maybe some of these remains are similar to 1 or 2 or 3 or grouped into our groups of nowadays populations. So the stages now are collection of archaeological grape remains, of course, morphological characterization, because most of our remains are charred, are burned. Okay, so the success rate of DNA sequencing of these remains shouldn't be very high. So we are going with two approaches, one on the morphological way and the other on the genetic way. And I'll explain it. The morphological characterization is done by measurements, as I will show in a few minutes, and by 3D analysis done by Avshalom Karasik here in the Hebrew University. And of course, aDNA extraction and sequencing, which is, we are starting it just now. So you must be patient yet. So we got a lot of archaeological remains. There are a lot of grape seeds going around in the archaeological sites if someone is actually looking and separating them. But I think that our most interesting finding, it's not mine of course, it's Dr. Ilat Mazar's finding, is whole grapes coming from the offel. This is the offel site, from jars like this one. And on this jar, they found an inscription in very, very ancient Hebrew written Yain Chalak. Yain Chalak is a smooth wine, which gives us a very good context. Okay, these grapes are probably in a way connected to wine. So we extracted, of course, seeds from these, which are very good preserved. They are, of course, burned, but they are very, very much whole. They are very nice. So we are analyzing all of these. Ehud Weiss and Itamar Gislev from the Bar Ilan University do the job of collecting the archaeological samples. And Avshalom Karasik, what we're doing with him now is we are very high resolution 3D profiling of the different varieties we have now today in Israel. After that, we will want to throw an unknown archaeological finding and trying to see if we can actually match them. There are a lot of epitopes on top of each seed, and if you look in the right resolution, you should have a good identification. So this is one part of the job we're doing. In order to do that, we have to know how the seed shrinks, because when it's burned, it doesn't stay in the same shape. As you can see here, these are seeds burned in different temperatures, from 150 degrees to 450 degrees, done by Oshrit here in the audience. And you can see that there is a very specific region in between 3 hours and 250 degrees and up to 3 hours and 350 degrees that most of our findings, archaeological findings, look in this shape. The rest are very, very let's say, unstable, breaking down very fast, or they are not totally charred. So there is a region, and Oshrit also gave it dimensions, because we want to know how it shrinks. Is the shrinkage identical in every dimension or not? And we found out that the thickness doesn't change much. What changes a lot is the breadth and the length of the seed. So this can actually, the thickness can give us a good reference to the initial size of the seed. Now, flow work for the DNA extraction. What we're actually doing, we extract DNA, we repair the DNA. The DNA is from charred seeds. We do an end repair, TA end repair, adapter ligation. Then, of course, PCR amplification. We put indexes in, and we throw it into Illumina HiSeq. This happens today. It's just flying to Etzfat today, our first analysis, so I don't have any results to show you yet. but we are very much hopeful about this possibility. We have interesting findings of desiccated seeds. This is the only desiccated seed remain from Timna. This is in Udi Weiss's laboratory. This might give us much better DNA quality, so that can give us a good result later. So we do have DNA, and it's actually in about 40-50 base pair size. And we actually even have a PCR result. So we do have something there. Now we have to send it and sequence it, and we'll see what we get. So in summary, we collected and genetically analyzed the initial Israeli grapevine germplasm. We identified varieties suitable for wine production and a lot of Silvestris too. We collected samples all over Israel of archaeological findings, and hopefully we will, let's say, we hope, we are looking forward towards possible identification. Then we might be able to tell people what was the wine that King David actually drank. So great thanks for the JNF for the funding and for the good people that are doing the job, collecting all over Israel and doing the job in the lab. Yaakov Enig, Hodaya, Oshrit, Batchen, and Avital. And for some of the collections in Israel for giving us access to the collection. Thank you.
Thank you very much. Are there any questions?
Very interesting. I have a question. Do we know the exact variety which were used at the time of the Jews, the Israelites, 10th century onto the Cephas? I would guess even, say, the 7th century, so it was still used like this. That's one question. Secondly, is it, I research history. So, yet my question stands, can you think, based on historical knowledge, or can you deduce through archaeology what is, do you need a text basically, or can you deduce by yourself?
So first of all, for the historic aspect, we do have historic findings from about the 4th century, naming varieties that are still used. This is a phrase from the Gemara. There are varieties that were still used in Jerusalem in that time, and we know the names to go down in history, and we find these names today. So there are historical findings that give us knowledge about which grapes were used for wine production. They say by themselves that these are not very good varieties, so these are not the varieties we are looking for. But for sure there was a very widespread wine industry, so they had probably a lot of varieties. And yes, I hope that through archaeological findings, morphological and DNA analysis, we should know at least the big groups that were used for wine production, if not specific identification. Not yet. Okay. This is Udi Weiss's part in the work. Hopefully, yes. Hopefully, we will work with everyone. I'm not sure they found grape seeds. They found remains of wines in jars. They actually found tartaric acid, which is, by the way, not a very good indicator for wine because it's in other stuff too. Yes, we are very much interested in working with everyone that has any kind of grape seed. Probably not. DNA doesn't react very good for ethanol, as you know. There is a kit of NextNab that does the DNA repair, trying to put it in a better condition for the sequencing.
Did you look at the wine growing in Christian monastery?
Yes.
Suppose the Islamic brood would not go and destroy all the in each monastery grape growing.
Actually, I did it very, very fast because of the time, but yes, one of our main, let's say, candidates for places that we can find the grapes were in the monasteries. But unfortunately, they were very smart guys, and they imported varieties from Italy and France. So they are using today varieties that are known for their good wines, not just in Israel, but all over the world because they want to sell. So this is, in fact, what's happening now, unfortunately. But we did not finish the work. By the way, we have more than 50 sativa accessions that are unique for Israel. That's a lot. So we actually have a lot to work with. And we hope that at the end of this year, we will be almost the double amount of this. So we will have a lot in our hands.
Another question.
So curiosity, I don't know too much about grape genetics, but has people shown gene flow between sativa and silvestris? I mean, is it possible that you were doing this kind of wide variety, which is quite rare? Is it silvestris who actually found it right? But this could have just been some sort of
Of course, there is. They're the same species, so they are fertile. There is the gene flow, so that's one of the good questions about our white sativa. It was found in a very secluded region, so I don't believe it saw in any possibility a sativa variety as an ancestor, but it could happen. And that, yes, there is a gene flow, strong gene flow, and that's part of the interesting part of this work.
Do you conclude that this is indigenous plant?
Let's say this is what I want to conclude. But I'm very, very careful about this. It seems like we have a separated population of Vitis vinifera sylvestris. Yes, indeed. So it may have been originated here, or it may have been originated very, very far away in history from somewhere else. But let's hope it's... we will have these answers, hopefully, when Hodaya will finish her master's degree in a year.
So we're looking forward to this.
Thank you
very much.